So let's start with this story. So this is a 911 call with a 42-year-old gentleman who was dizzy and was sweating a lot. And what it says there was a stretcher was brought into the residence. The patient was getting ready for transfer from the chair to the stretcher when he started posturing and having a seizure. And I think so then he was transported to the unit, which is the ambulance, or from the house to the unit. And once in the ambulance, he went into cardiac arrest, and this was the rhythm. So it's ventricular fibrillation. So it's a very chaotic rhythm of the heart, the lower chambers of the heart, and was resuscitated from this. So this is shocked out of this, and subsequently had this pattern, which is a sign of an acute heart attack. So the patient basically collapsed due to this heart rhythm, resuscitated, had an acute heart attack, went in the hospital, had an artery that was blocked opened, and had a stent placed. But given all that went on up here in terms of had suffered brain injury and died about 10 days after being in the hospital for about 10 days. So this, unfortunately, this scenario of sudden cardiac death is quite common. It's the leading cause of death in the United States. And if you think about why people develop this problem, why people develop heart attack, there are several risk factors that we've known about for many years. And here are this patient's risk factors. So if you look at the LDL cholesterol, so-called bad cholesterol, HDL cholesterol, triglycerides, and all the other factors, the ones that are abnormal or slightly abnormal are shown in red. So the patient had above average LDL. So the population average in the United States is 130. So his is 167. The HDL is a bit low. Population average is like 50 or above for men, and is 40. The triglycerides are a little high, and then there's also family history where the father had a heart attack at a young age. So this kind of presentation, I think, highlights three questions that I'll try to get through today. One is, what is the inherited basis? What's the genetic basis for early heart attack? The second question is, beyond the bad cholesterol, the LDL cholesterol, what lipid pathways cause heart attacks? I highlighted two other ones, the HDL and the triglycerides, so we'll talk about that. And then lastly, are there things that lead to heart attack beyond the cholesterol, beyond lipids, non-lipid pathways to heart attack? Okay? So let's just get everybody up to speed on this problem. So the medical term, of course, is myocardial infarction, so that's commonly referred to as heart attack. This problem is a problem of the arteries that supply blood to the heart. These arteries run on the surface of the heart, and if you slice them open like it's shown up there, over time what happens to these arteries is in the wall of the artery there's buildup of plaque. That's the yellow stuff that you see there. And that plaque it takes about 20, 30 years for that to kind of progressively build up in the average person. So there's a chronic phase of this disease that takes decades for the plaque to develop. And then there's an acute phase where the plaque bursts open and the flowing blood is exposed to the collagen underneath the lining of the wall of the artery, interior lining, and that's a very potent trigger for blood clot to form. So acutely a blood clot can form on the side of the plaque, and if that blood clot blocks blood flow for more than 20 minutes, then the heart muscle here, for example, that's served by this artery, that heart muscle can die. That death of heart muscle is a heart attack, is myocardial necrosis. And that death of heart muscle can lead to certain things. One is the chest pain that people develop in the setting of a heart attack. It can also lead to characteristic changes on the electrocardiogram, the surface electrocardiogram. And then some of the proteins in the heart muscle cells can leak out. And you can measure that in the blood, and that's a myocardial necrosis, and that's part and parcel of the disease as well. So this disease is a classic, what's called a complex trait. So that's a disease that has both a genetic component, a heritable component, as well as lifestyle component. And about half of all heart attacks present like that patient that I just told you about. The first presentation is sudden death. One other background information is we've known for maybe 50, 60 years, actually even more, that there are some risk factors, lipid risk factors for heart attack. Shown up at the top is cholesterol. On the bottom is triacylglycerol. These are both essential items in terms of the body for cells to function properly. But this stuff doesn't dissolve in water or dissolve in blood. So the body needs a transport system for these molecules, cholesterol and triacylglycerol, and that transport system is called lipoproteins. So these are macromolecules that look like balls, essentially. They're spheres, and in the middle is the water-insoluble stuff, the cholesterol or the triglycerides. Triglyceride in blue and cholesterol in orange. And then there are different types of lipoproteins. The HDL, the so-called good cholesterol, that's shown on the left there. It's small, it has high density, and it has a certain protein on the surface called ApoA1, and then mostly cholesterol in the center. These are all other lipoproteins. LDL, so-called low-density lipoprotein. And then these guys here are all what are called triglyceride-rich lipoproteins. They mostly have triglycerides in the center, the blue. So that's a little bit of background on lipoproteins. Okay, so let's get into the problem then in terms of the genetic causes of heart disease. So why does somebody develop a heart attack at a young age? So to understand the inherited basis for early heart attack, we began going back to about 1997, enrolling patients at my hospital, Mass General Hospital, with heart attack at a young age. So these are men less than 50, women less than 60 was our criteria in the very beginning. And so if you think about the distribution of age at heart attack, the average age of those who have a heart attack in the United States for a man is 66 and a woman is 71. And so people who have heart attack in the 40s and 30s, they're in the one end of the bell curve, right? So there are two major reasons you can think about why somebody might have the genetic causes of early heart attack. The one model is called a monogenic model, where a mutation in a single gene has a large effect. It's sufficient to lead to early disease. The other model is called a polygenic model, where the additive effect of many variants leads to early disease. Okay, so I'm going to describe to you results from our studies to look at these two models for heart attack. So the monogenic model, the study design is shown here. We did a study a few years ago where we took about 5,000 cases, so people with heart attack at a young age, and 5,000 controls, and we sequenced every gene in the genome, exome sequencing, in all 10,000 individuals, and compared the rate of mutations in cases compared to controls, with the idea that a risk gene, there would be more mutations in cases than controls. And we looked at gene 1 all the way to gene 18,000. And there are 4 signals that have emerged from these studies for monogenic causes of early heart attack. And these are the 4 genes. One is low-density lipoprotein receptor, another one is lipoprotein lipase, apolipoprotein A5, and then apolipoprotein A. What I'm showing you here are the frequency of the mutation. So in the population, for example, for the top one, 1 in 200 people will carry a mutation in that gene. And that gene will, as a result of carrying the mutation, they'll have high levels of LDL, and then they're at higher risk for heart attack, about 3 to 4-fold increased risk. For these 2 genes, here's the frequency. Here, the elevated fraction, lipid fraction, is triglycerides, and they're at higher risk, this level of risk, compared to people who don't carry the mutation. And then for LPA, it's shown at the bottom. So these are the 4 genes where coding mutations, so mutations in the coding sequence of a gene, confer large effect on MI risk.
And then one
Yes?
individual can have mutations in multiple genes? Or
Well, here, no, because it's so rare. So the probability of having this and this is the product of the 2. So it's going to be pretty rare. But so in general, if a patient has a mutation, it will just be in one of these genes. Okay, so that's the monogenic model. You can essentially say that if you carry a mutation, if you're among the 1 in 200, your risk for having a heart attack is 3-fold higher compared to the other 199 out of 200 that don't carry the mutation. So what that means, though, in terms of, over a 10-year period, for example, if you took I don't know, 10,000 people, you got all in every single person you had the sequence data. So if you had 10,000 people you sequenced, you roughly would find, 50 people would carry a mutation in this gene, and the remainder wouldn't. Is my math right? Yeah, 50. So, and then you want to say, what's the heart attack rate in the 50 compared to the remainder? What I'm saying here is the heart attack rate in the 50 would be threefold higher compared to everybody else. Now, what exactly that rate is basically depends on the age of the population, the 10,000 people. So roughly, it would be something like 5%, let's just say, over a 10-year period, of the non-mutation carriers would have heart attack, and in that same frame, then the mutation carriers would have a 15% risk of heart attack over the 10 years. So that kind of prediction is possible, and this is the level of stratification you get between. Now, one thing to note here, which I think it's sometimes a little hard for people to wrap their head around in terms of genetics, because most people automatically equate genetics with determinism, that they're basically, if you have the mutation, you're going to have the problem. But you can see here that's not the case at all for this disease. There are many people who will have a mutation and will not have a heart attack over a prolonged period of time. So here, kind of, there's not a one-to-one correlation between genotype, the mutation, and phenotype, unlike some other diseases, some other genetic diseases.
Are these all heterozygous?
Yes, these are all heterozygous. Now, you can get this in homozygous form, and those individuals are extraordinarily high risk for heart disease. There it's almost a one-to-one correlation. It's an additive. And these have also been seen in homozygous form as well. But you can imagine the frequency of people in the population with homozygous is more like 1 in 300,000 or so.
Does these confer any other advantage to these mutations?
No, these are all risk genes, so yeah, there's no known benefit for these. Okay, so what you can see here then is, if you take 5,000 people with heart attack, as we did here, right? Early heart attack, and then you sequence them all, you basically only find an answer, quote unquote, in the coding sequence in 2%, in small fraction of the, some 1 to 2%. So that's like 100 people. So why do the other 4,900 have a heart attack at a young age? So we've explored this other model, right, a polygenic model, the idea that, again, additive effect of many variants. And so the study design here is you take people with disease, again, without disease, and now the sample sizes are much larger because here we're not doing sequencing, but we're doing genotyping chips, arrays like Affy or Illumina. And now we're not looking at a gene by gene, but we're looking at polymorphism by polymorphism. So the unit of analysis is not the gene, but variant by variant. And we're looking at millions of variants and asking at variant 1, variant 2, variant 3, at each variant, what is the frequency of the allele in cases compared to controls, and asking if that exceeds a significant threshold that's pretty stringent for all the multiple testing. So the results of these kind of analyses, these are genome-wide association studies, can be summarized here. There's several of them we've done over the years for heart attack, and on the x-axis are the chromosomes, on the y-axis the strength of statistical significance, and any dot above this line is considered significant, and each dot here represents a specific spot in the genome, a specific variant, specific polymorphism. And the little labels are genes nearby, and you can see there's a lot of different dots that exceed the significance threshold. So right now there are about 95 specific letters in the DNA sequence that exceed this stringent threshold and are associated with coronary disease. Okay, so that's kind of an example of how the polygenic data would pan out. So you could now ask, if you have 95 spots in the genome that relate to coronary disease, how many of them actually relate to blood cholesterol levels? And it turns out there's about 15 of them that relate to cholesterol levels. But the other 80 don't relate to cholesterol. They're non-lipid, let's say. So it gives you a sense of, like, there's a lot out here that could play an important role for heart disease beyond what we already know about, which is lipids.
But these are not in, all are not in the coding sequence.
These are all almost all non-coding sequence. So these labels of the genes are just labels of convenience. We don't often know for sure that this is the right gene even. They're all regulatory, exactly. There's some small fraction of these are coding as well, but most of them are non-coding. So one thing that is interesting is this idea of prediction, getting back to, but using the entire feature set. And by feature set, I mean, instead of just relying on the top polymorphisms here, can we take advantage of everything? Okay. So this idea of polygenic scores goes back a few years. We first showed this in 2008, that you could take, let's say you had a certain number of polymorphisms related to disease, and so a person would have 0, 1, or 2 copies of the risk flavor, and then you can have a score ranging from 0 to n. So for the 95 SNPs, you could make a score out of the 95 SNPs. You have a score of 0 to 190. And you could weight that score based on the effect size of the variant or unweighted. And we showed that that kind of scoring can work and in terms of risk prediction in a couple of different papers over the years. Most recently, we've extended this to look at this idea of, if you go beyond this 95, what if you take advantage of all this information? Could you do a better job in terms of prediction? So I'm just going to show you that quickly. So these polygenic scores now are moving to, actually this one that we've developed moves to all 6.6 million variants across the genome, measured in each person. And we make a score for each person based on the 6.6 million. And the hypothesis here that this kind of score can actually now maybe identify individuals of risk equivalent to those monogenic mutations I showed you earlier from LDL receptor and so forth. So the study design here is we have a completed genome-wide association study that has a list of SNPs and a list of effect sizes for each variant. We took that and we tried several different computational algorithms to derive scores, and then we tested in a test data set to say, in a derivation data set to say which score is the best. And this was the first part of UK Biobank, about 125,000 people. And then we picked one score that came out best here and then applied it to the rest of UK Biobank. And two things to note, this is the score calculated in about 300,000 people in this large prospective cohort study called UK Biobank, a research study. And so beautiful bell curve. So this is essentially a quantitative score for your inherited liability to heart attack. So this looks like many other traits in the population, like height or cholesterol. So then you naturally wonder, like, what if you happen to be on this end of the bell curve, the risk end of the bell curve? What is your risk compared to everybody else? So you can now ask these kind of questions with this quantitative distribution. Actually, before I show you that, the other point to make here is you have now a quantitative score of risk. How does this score compare to known risk factors for heart disease, like cholesterol or smoking or blood pressure? There are existing risk prediction algorithms using those risk factors, and one of them is called the ACC-AHA pooled cohorts equation. This is used by doctors regularly to predict somebody's risk of heart attack, and it's based on just some clinical factors, actually those risk factors that I put up on the slide in the very beginning about that patient. What you can see here is that the correlation coefficient of this genetic liability is almost nothing with the existing, very low with the existing algorithm. So this is capturing a dimension that's of risk that's orthogonal to the existing information that's used. So now we can ask, what if you happen to be in the top end of the bell curve? How does your risk compare to everybody else? And here's the answer to that. So if you take the top 20% of the distribution compared to the remaining 80%, the risk is here. But I want to draw your attention to this. If you take the top 5% of the distribution and compare to everybody else, your risk is almost fourfold. So this is very similar to the LDL receptor mutation that I just showed you earlier. But the main difference is that that was relevant in only 1 in 200 in the population. This is relevant in 1 in 20. right? So a lot of why people are having heart attack is this polygenic, early heart attack is this polygenic basis. So, I mean, another way to think about it, I mean, just instead of, focusing on the specifics, you could just say, look, it's just a number that tells you your genetic liability to heart attack, like your cholesterol level tells you about your risk of heart attack. And it turns out that, this number, as I said, it's a pretty good predictor. Now, one other point to make here is that all this work that I just showed you is in individuals of European ancestry, so in whites, because 95% of the UK Biobank is white. So this, but allele frequencies differ across ancestries, and the effect of a given variant might differ across ancestries. So how relevant is this score that I just showed you for populations outside of Europe? So, this part of the world. So, and we did this we're working on this in collaboration with Sam, but this is data from the UK Biobank, where there are about 7,500 people of Asian Indian ancestry. And it turns out the polygenic score has the same distribution in these Asian Indians. And then the risk gradients are actually quite good. They're basically almost identical to the whites. So the score developed largely from data in whites, at least for South Asians, looks pretty good. And it turns out that we've looked at this in blacks, Hispanics, East Asians, and it's not as good actually for those ancestries. You could imagine, basically the score is less and less effective as your ancestry goes further and further away from Europe. It turns out South Asians are about 60% of European ancestry, but Africans are almost zero. So it's but in this group at least it's generalizable. So to summarize then, the monogenic and polygenic models, what I've tried to show you is that the monogenic model prevalence is about 1 in 200. The odds ratio for MI, you can detect using LDL cholesterol or DNA sequencing, and then the LDL reduction, the interventions are LDL reduction and risk factors, control risk factors. The polygenic model is much more relevant and relevant in a larger fraction of the population, similar effect size, but these individuals are currently undiagnosed. They don't stand out in terms of their traditional risk factors. And the question then is, if you do find these individuals, what could you do? Can you modify that inherited risk? Right? Are you doomed to have the problem? And so we've looked at this, and so is polygenic risk for MI modifiable? And I won't go into the data here, but we've shown that with lifestyle, adherence to a healthy lifestyle, you can reduce the inherited risk by about 50%. And also with medicines like statin medications, lower the cholesterol, that also lowers the genetic inherited risk. So for this disease, the genetic risk is not, DNA is not destiny, it's modifiable. So here's the summary then. Monogenic risk, if you had 100 people with early MI, you can explain it probably in 2 with monogenic risk, a much larger number with polygenic risk. And there are some unfortunate souls who actually have both monogenic and polygenic, and they're much higher risk for a disease. So remember I showed you earlier the correlation with the existing, the ACC-AHA risk score? And that correlation coefficient was like 0.03. So that immediately tells you basically that these patients, their risk factor profiles, if you compare the high genetic risk versus everybody else, the traditional risk factor profiles are quite similar. So they do have a little higher blood pressure, they do have a little higher LDL, they do have a little higher body weight, but it's not off the charts. You wouldn't pick them out of a room and saying, oh, that's the reason you're going to have a heart attack, because your blood pressure is through the roof. So does that answer your question? So, because we've done this where we've actually, we've calculated you can take this top 5% of people and the 95% and make a table of the traditional risk factors in the 2 groups. And they're basically largely similar. That's why I'm saying that these people are unrecognized right now. Because they're in their 30s and 40s, their risk factors don't look that abnormal, and their doctors are not doing much about it. But their first presentation might be like the patient I showed you. Yes, we've looked most closely at the people that are young, but it turns out that there's no magic between the less than 50-year-old heart attacks and the 55-year-old with heart attack. It's basically just a matter of effect size. So these gene variants have a larger effect in terms of early disease, and the effect size diminishes with later disease, presumably because of the larger role of environment as you live longer. Okay, so beyond LDL, what lipid pathways cause heart attack? So remember, I showed you the patient had slightly abnormal three things: LDL, HDL, triglycerides. And when we think about things measurable in the blood and ask how those things, do those things cause a problem like heart attack, the traditional approach we take is what's called observational epidemiology, right? X-axis is the risk factor. On the y-axis is the heart attack risk. And you have a correlation, positive correlation for LDL, negative correlation for HDL, positive correlation for triglycerides. So when you see something like this, people say, oh, LDL is correlated with MI risk, therefore LDL must, the assumption is LDL causes heart attack. But as you know, this doesn't mean, when two variables are correlated with each other in the population, it doesn't mean one causes another. It could be the two variables are correlated because one causes another, or that the heart disease, the outcome causes the variable to change. So that's called reverse causation. Or there could be some middle factor, a confounder that relates both to the exposure and the outcome, and that's the true source of the association. So it's actually very challenging to distinguish this cause from mere correlation when you just use observational epidemiology. And we get in trouble like this all the time because you see every day like eating walnuts is associated with X, Y, Z, or chocolate is good for you, bad for you, blah, blah, blah. All those are correlation studies just like this, X and Y, and very few of those actually reflect causal relationships. And how do you get to cause, and how do you distinguish cause from it? There are basically just two approaches in humans. One is human genetics, the other is treatment. The definitive is actually you try to change that, right, LDL, and then the disease risk changes. Then you've proven that is a cause of a disease. But it's very expensive to do these treatment trials, and so you want to have some idea ahead of time. And so human genetics can give you a clue as to whether a biomarker is causal or not. And the idea is very simple. Like if LDL caused heart attack, then gene mutations that raise LDL lifelong should increase risk of heart attack. And conversely, gene mutations that lower LDL should lower risk of heart attack. That's the idea. So it turns out for both treatment and genetics, LDL has been proven beyond doubt to be a causal factor for coronary heart disease, for heart attack. And you have statin medications, ezetimibe, this new medication, all these lower LDL, they lower risk of heart attack. And then you have a bunch of genetic evidence, like I just showed you, the LDL receptor mutations that raise LDL, increase risk of heart attack. Now, in addition to medicines the mutations that increase risk, we've had this whole story now about PCSK9, which you may be familiar with. Here's the opposite. Here's a mutation that doesn't increase risk, but actually lowered risk for disease. 1 in 50 people carried a mutation in this gene. They had lower LDL cholesterol lifelong, and they had lower risk of heart attack. And this was very useful because it suggested that if you could develop a medicine that mimicked this mutation, then it would be helpful to people in general. And that's what happened. These companies developed medicines that block the PCSK9, basically mimic the natural condition, and these medicines have now been proven to lower LDL and lower risk of MI. So we have very conclusive evidence that LDL is a causal factor, both from the risk side and the protection side. So for HDL, believe it or not, so even though it's called the good cholesterol, I'm sure all of you have been told that your level of good cholesterol, it actually it's not clear yet whether that's a causal relationship or not. So what I mean by that is, does your high HDL, does it actually causally protect you? Or is it actually just marking something else? Is that, you follow? Okay. So we decided to look at it from a genetic perspective. Again, the idea is if HDL was causal, then a gene mutation that raises HDL cholesterol, the people who carry that mutation, they should be protected from risk of heart attack, right? So here's a gene mutation that raised HDL cholesterol. This is in this gene, LIPG. 1 in 40 in the US carry a mutation of this gene. As a result of carrying the mutation, they have about 15% higher HDL compared to the 39 out of 40 that don't carry the mutation. So we wondered whether these people that carry the mutation, are they at lower risk for heart attack, as you would expect if HDL was causal. So we tested the relationship of the mutation to heart disease risk, heart attack risk, in a little over 100,000 people. This is all the data from the different studies. And then here's the summary odds ratio, 0.99. So this was very surprising to us when we first saw this. I actually had a lot of trouble getting this published. So this suggests that HDL is not a causal factor, that despite the fact these individuals have lifelong higher HDL cholesterol, the mutation carriers, they have the same risk of heart attack as those who didn't. This correlation is very robust in the population, but the actual reason why people with high HDL have lower heart attack is because of something else, right? Doesn't have anything, it's not the HDL itself. Okay, so these data would suggest, right, that a medicine that's designed to raise somebody's low HDL level, right? There are many companies that for many years have been, again, the idea is like high level is correlated with good things, low level is correlated with bad things. So if your level is low, let's try to raise it, right? So you have a low level of the good cholesterol. I'm going to try to develop a medicine that raises it with the expectation that would reduce risk of heart attack. But our genetic data would suggest that HDL is not a causal factor, and therefore an attempt to raise the HDL would not work. Okay, so we predicted that medicines to raise HDL would not work. Okay, so it turns out, actually just several weeks or maybe a couple of months after that paper was published in 2012, this result came out. Okay, this is a medicine in a randomized controlled trial called dalcetrapib that raises HDL by inhibiting this specific protein. Okay, these are results from the trial where this is the HDL on the y-axis and time on the x-axis. And this is the treated individuals with the medicine and untreated. And the medicine raised HDL by about 30%, 40%. So you give this drug, and I think this trial was about 12,000 people. So half the people got the medicine, half didn't. The half that got the medicine had markedly higher HDL, developed markedly higher HDL, and compared to the placebo. So what happened to their heart disease rates over time? Here are the heart disease rates in the two groups. Completely superimposable Kaplan-Meier curves, right? This is not what you want to see at the end of a 12,000 patient, $300 million trial. So, so I think that this is additional evidence, and there's now several other HDL raising medicines that have failed in the clinic, failed in randomized controlled trials. So the old view, I think, is HDL cholesterol is good cholesterol, but the new emerging view that it's a non-causal marker of risk. So, but I think one important point to make here, you go to your doctor, you get your HDL cholesterol checked, right? Should you still be checking HDL cholesterol? Absolutely. Because it's very important to distinguish between a biomarker and its usefulness in predicting versus utility in terms of treatment. So a non-causal factor like HDL, a non-causal biomarker is perfectly useful, can be perfectly useful for prediction. But it's only causal biomarkers that you want to approach with treatment with the expectation that that would reduce risk. And the silly example I like to give is like gray hair. right? So if you do on the x-axis the proportion of hair that's gray, on the y-axis risk for MI, there's a linear relationship in the population. So the more gray hair you have, higher risk of heart attack. But of course, gray hair, sorry?
It's also age.
Well, yeah, age is a confounder. So gray hair is not a causal factor. I mean, dyeing somebody's gray hair black wouldn't reduce the risk of heart attack. So there's confounders. And so, but gray hair could be a very useful predictor. So if I, had a room of 1,000 people, and I ask you to just look at them and then tell me who's the highest risk for heart attack. If you took all the gray-haired people and put them on one side of the room and everybody else on the other side of the room, you'd do a pretty good job of risk prediction for heart attack risk. Because you would have picked out all the old people into one side, and So gray hair, though non-causal for heart disease, can be a very useful predictor. So HDL is still a very useful predictor. If you have low HDL cholesterol, you are at increased risk for MI. It's just not because of the HDL cholesterol. So now, one question that comes up then is if you have this relationship between HDL and MI risk, and I told you it's not a causal relationship, then what is it? Why do people who have high HDL, for example, have lower MI risk? Now, it could be this confounder, like it could be not a confounder, but another variable that both basically can explain this and this so-called intermediate variable, right? And it turns out that high HDL tracks with many other things, like low triglycerides, or being lean, or being very physically active, all these other potential factors that could truly be the cause of the lower MI risk. So we've tried to explore whether maybe it's the low triglycerides that tracks with a high HDL that is actually the causal part of it. So, so this is a story about the Lancaster Amish. So these are immigrants to the United States from Switzerland from the 1700s. A few hundred people came over. And then they've stayed a closed community in a couple of different parts of Pennsylvania. And there are now about 100,000 of them living in outside of Philadelphia. And we found several years ago that these individuals, 1 in 20 of them, have a mutation in a gene called apolipoprotein C3 or ApoC3. And actually 1 in 150 in the United States have the same mutation. And this is a mutation that breaks the gene. And here, just like the PCSK9 story, these people that carry the mutation, they're not at higher risk for disease, but they're lower risk. They have lower level of blood triglycerides and lower risk for heart disease. So this is a pretty interesting observation. It was an interesting observation because it suggested that if you could block this, if you could mimic the mutation, you might have a new drug, basically. Now, this is just having one mutation, one copy gone. What about if you had both copies gone? Would that be bad for you, or even better than just having one copy gone, right? So there's this idea of humans who have one copy of a gene broken. That's a gene there producing protein. One copy of the two copies gone, heterozygous null. We found that for ApoC3. What about if both gone? This is called homozygous null. These are also called knockouts, human knockouts, like a mouse knockout. Complete disruption of a gene, but it's natural. Can you find this? Why would we want to find people who lack ApoC3? Well, it suggests that you could completely get rid of the gene and it would be safe, right? Pharmacologic inhibition might be safe if you could find a homozygous null that actually was healthy. So we were trying to find the homozygous nulls, and it turns out you can take advantage of a specific property of human genetics, which is that when parents are closely related to each other, the concept is called consanguinity, then the children are more likely to be homozygous in large stretches of the genome. Okay? This is a map of first cousin marriage in the world, around the world, rates, and color-coded. The darker colors means more of the marriages are. And you can see that a lot of North Africa, Middle East, some of southern India, there's a lot there's some consanguinity. It turns out the highest rate in the world in terms of a map of consanguinity is in Pakistan. And we had a collaborator who worked in Pakistan and had a cohort study called PROMISE that had about 7,000 people that had been recruited, where a lot of them there's a more than 40% of the marriages were first cousin. So and we looked at that cohort, and in that cohort we found the world's first ApoC3 homozygous nulls. ApoC3 is measurable in the circulation. It's a circulating protein. And on the x-axis here are the genotypes, plus, 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 minus, minus, minus. So these are the homozygous nulls. On the y-axis are the ApoC3 levels. What you can see is there are 4 people who basically have no circulating ApoC3. Okay. So we were surprised but pleased to see this. And then we had one, this is all Danish's work. He's from Pakistan, and he did all this work. So he recruited one of these people, one of these 4, and then got the rest of that person's family. So there's the proband, that's homozygous null. There's the spouse. They had 9 children, which was very good for human genetics. And and this little line means that they're cousins. Okay, so we were shocked to see that the spouse was also homozygous null for the same gene. So that means all 9 children have no circulating ApoC3 as well. So it's a pretty unique family in the world. And so now we have 11 individuals in one family that have no circulating ApoC3. Turns out there's actually a larger part to the family. And in this family, you can find all 3 genotypes, plus, 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 minus, minus, minus. So then we decided to say, maybe can we learn some biology based on this? So why are the people who have ApoC3, this gene broken, why are they protected from heart attack? They have lower triglyceride levels. As I said earlier, they are protected from heart attack. And we know what this gene does, or it's been known for a while, that it puts a brake on your body's ability to digest dietary fat or clear dietary fat. So this is a bad guy. It puts a brake on your ability to clear dietary fat. So what we decided to do was a challenge for these 2 groups, with and without the mutation. We gave them a dietary fat challenge, basically a big milkshake, about 80 grams of fat. And then we measured blood for every hour for 6 hours afterwards in people who are homozygous null versus the wild type in the same family. So, and here's the data in terms of the dietary challenge results, where on the y-axis is the blood triglycerides, the blood fat level. On the x-axis is time, 0 to 6 hours. And this is the wild type individual. So presumably all of us in this room, after you eat a fatty meal, your blood triglycerides goes up very quickly within 6 hours. Almost doubles 2.5 times. This is happening with every meal, by the way, in your life. But look what's happening in the homozygous null individuals. So their blood triglycerides doesn't go up at all, doesn't budge. So with a lifetime of meals, we are all seeing, our arteries are all seeing the fat. These guys are not seeing the dietary fat. So you can now have an understanding of why they may be protected from heart disease.
And no side effects.
Yeah, there's no, they're otherwise healthy.
So why is this mutation so uncommon?
Well, okay, I can answer that because there's been, over time, heart disease, for example, was not an evolutionary pressure. Right? It's really a disease of, it's really, for most of humanity, undernutrition was the problem. And coronary disease is basically a disease of excess nutrition, right? And it's only a problem in the last 200 years. Most of humanity, the major selective pressure was basically likely infection and lack of food, undernutrition. And so we don't exactly know, the other potential consequences, the full spectrum of consequences of lacking the gene. There may be something that's bad that we haven't seen, but in these, right? Don't. But in this family, in this village, there's been nothing seen.
So how does the heterozygote look like? Will it be in the middle?
Middle, yeah, exactly in the middle, yeah.
These should all go into a marriage dossier.
Right, exactly.
It was the only.
Exactly.
Did you look at HDL and LDL as well?
Yeah, so they do have, these guys do have they have higher, so they have low triglycerides, they have slightly higher HDL, and they have, their LDLs don't change, differ much. But this is a gene that affects both, like several of the other genes I mentioned. Yeah. Okay, so now this all suggests then, can we develop a medicine, right? We think that the reason that we think that HDL is a non-causal biomarker of risk, and what it's marking is actually the level of triglycerides and actually your body's ability to clear dietary fat. And that it's actually the triglycerides in that dyad that's the causal part. And the missing piece here is that what you'd like to see is medicines that predominantly lower the triglycerides and lower risk of heart attack. And that's where we are right now. There are several companies testing medicines that lower triglycerides to see if they lower risk of heart attack. And one of the triglyceride lowering medicines is an antisense oligonucleotide that destroys this gene, ApoC3. And that's already been shown to lower triglycerides, but the heart attack studies are still pending. Maybe I should just take 2 seconds to just tell this story. There's another gene, ANGPTL3. This will be quick. So here it starts with this woman, Anna Fuhrer, who works for this company, Purina, back in the 90s. She went to a corporate health fair, had this lipid profile. And, this is enviable, right? 1937. And the HDL is very low, but I just told you that may not matter. But her doctors are very concerned. They thought this might be very bad for her because it's too low. So they referred her to a lipid specialist, Gustav Schoenfeld in Washington, St. Louis, and he brought the entire family in. Again, 3 generations in living in St. Louis. And this is Anna right here. And this, her mother and her uncle and several of her siblings had the similar lipid profile, low, all 3 lipid fractions being low. We worked with Gus to solve the problem in terms of what gene might be responsible, because it looked like a single gene was the reason for the low lipid levels. And we found that the single gene was ANGPTL3. And it's a very interesting pattern here. So there are 2 copies of ANGPTL3, and there's one mutation running in the family called S17X. So this is a stop codon in the first exon. And Anna's mother, a couple of her uncles had this mutation, and she inherited this mutation from her mother, okay, as well as several of her brothers and one of her sisters. Okay, but what was really fascinating about this family is the father, see here, married into this family with a different nonsense mutation in the same gene. Okay, and they also had 9 children, so also very good for human genetics. And these 4 people inherited 1 broken copy from the mother and 1 from the father. And the 4 in the family are basically human knockouts of ANGPTL3. And these 4 are the ones that have this like terrifically low lipid levels for all 3 lipid fractions. So what this suggested was that here's another gene that could be completely gotten rid of, and has a very good lipid profile, right? But then the follow-on question is, are these people protected from heart disease? Are they protected from heart attack? And we've gone on to look at that in 2 ways. One is we brought Anna and a couple of her siblings who are the knockouts, and then other siblings in the family who are wild type, and did this test, this CT angiogram test, where you inject in dye and then take X-ray pictures of the heart to look at the heart arteries. So this is Anna, actually. Her arteries are completely clean. And this is one of her sister, who's wild type. And she had a plaque. So 2 out of the 3 siblings that were wild type had plaque. 0 out of the 3 that are loss of function, complete loss of function. So homozygous knockout had plaque. So this suggested, hey, it is protective, but this is very anecdotal. Like, it's a comparison of 2 out of 3 versus 0 out of 3. So we ended up sequencing this gene in a lot of people, over 100,000 people. And in the heterozygous state, the null mutation was protective for heart attack. So here's another situation where heterozygous null, protective for heart attack, homozygous null, viable, healthy. So maybe we should develop a medicine to mimic this mutation. So there have been a couple of companies, Regeneron and Ionis. This is monoclonal antibody on the left, this is antisense, that use this target. And these medicines have been given to people, and they lower triglycerides, but the heart attack studies are still pending. So those are two stories, and in total now, we've identified, in the last column here is Decode Genetics, 7 protective mutation stories. And each of these, basically, there's some drug either available already or in development. So this is a general paradigm of using human genetics to identify targets, specifically human genetics, genes where there are mutations that, instead of putting people at risk for disease, actually protect against disease. And there's data of this type, not just for heart attack, but other diseases like Alzheimer's and you name it, a range of diseases. And I think this might be a very fruitful way to figure out which targets to go after for medicines. The other interesting thing is that beyond LDL, these 3 of the genes here, so 3 of these genes are all in the same pathway. They all work in the same pathway, basically to help your body clear dietary triglycerides. And they all function around this gene called lipoprotein lipase, which is a rate limiting enzyme in your body's ability to hydrolyze dietary fat, hydrolyze triacylglycerol. So beyond LDL, the key pathway for protective for heart attack seems to be not HDL, but rather this lipolysis pathway. Okay. That's the second question. The last non-lipid, I'll just go to the punchline. We identified somatic mutations, so the question earlier, in blood cells. And it looks like this is going to be a new mechanism for heart disease. So it's not germline, but somatic mutations in hematopoietic stem cells. And we showed that carrying these mutations increases risk for disease, both early heart attack and later heart attack. And the mechanism, about 2 to 3% of early MI cases carry these clones. And the mechanism seems to be increased inflammation. And this is data from one of the genes mutated, TET2, that shows that there's more atherosclerosis in mice, and then there's also heightened inflammation. So I won't go into the details here. All this is published. Okay, so what is the inherited basis for heart attack? I talked about both the monogenic model, but I think the new information is that the genome-wide polygenic score is a new risk factor for heart attack. Beyond LDL, what pathways? I talked about ApoC3, ANGPTL3, and triglyceride-rich lipoproteins as being important. And then we didn't have time to go into this, but the non-lipid pathway that we identified recently revolves around inflammation, specifically clonal hematopoiesis. Okay, let me stop there. Thank you.